Hello, everyone. My name is Hua Li. I'm a research assistant professor working at Department of Surgery, Watui Medical Center, Uniform Service University of Health Science.
Hi, I'm Sharad. I'm assistant professor of surgery at Uniform Service University of Health Sciences. I have the pleasure to work with my colleague, Dr. Hua Li, and I have the opportunity to work with him from last 10 years. We are working together on a similar project since I joined Center for Prostate Disease Research program, which is a translational research program of Uniform Services University of Health Sciences. And we are in a translational research team and working towards translational and basic research program of prostate cancer.
Okay, so we're glad to talk about our newly published manuscript, and the title is the characterization of unique PMEP1 gene plus variants isoforms C and E from RNA sequencing provide a novel insights into the prognostic evaluation of prostate cancer. So briefly, we just want to give some introduction why we want to do the work. And so for the most challenging in the field of prostate cancer is just the castration resistance and the metastatic prostate cancer. So it's very hard to be cured and it can cause the death of the patient. Although the incidence rates of prostate cancer has increased in recent years, but there is challenging from the hormone resistance and mass still is a major topic and a major headache for the researchers. So, and our lab start investigating the androgen signaling and the TGF-beta signaling and what's its function and what's the role of dysfunction of the two signaling pathways in the later stage of prostate cancer. And during this process, we found a gene we call the PMEPA1, stands for prostate transmembrane protein 1. And we found it can function as the important regulator of the both androgen signaling pathway and TGF-beta signaling pathway. So we mainly focused on is to function to degraded androgen receptor protein, subsequently leading to inhibition of androgen signaling. But this gene also another hot topic in other non-prostate solid tumors. The report is highly correlated with TGF-beta signaling. So we think about maybe this gene can function under some context, either in air signaling or TGF-beta signaling, and they can play an important role in the development, initiation, and the progression of disease into castrating resistance and metastasis. So, we already report ABC isoforms, 3D isoform was a gene before, is also reported by other reporters. But luckily, we used different sequencing, RNA sequencing, and also the protein alignment technology. So we discovered another important isoform. Although it's the low expressed PMP1E isoform, but when we look at closer, we dissect its function in terms like response to androgen, and also can promote the androgen independent cells growth to grow. So this is important for the potential application either for biomarker or the further elucidation of the underlying biology of the castration resistance and metastasis. So Dr. Sharad will give more detail on how we do this and how we overcome all the challenges during the experiments.
Yeah. So basically, thank you, Dr. Lee, for giving a very clear overview to the viewers. The idea when I joined CPDR, this PMP1 gene already been discovered. So I want to shed some light towards that this gene, which is known as prostate transmembrane protein androgen induced 1, has been discovered in CPDR in the context of prostate cancer. And during the process of they're looking for highly androgen inducible genes, and this gene has been popped up as a highly introduced by androgen receptor. So from there, we started looking about the role of this gene in prostate cancer context. And we have published several papers along those lines. We have a grant on the NIH R01 grant also towards these lines. The bottom line, when we look into that, this is not only the prostate cancer, but similarly, when we discovered in 2000, there are certain other groups who are working on the solid tumors. They also found similar genes which is inducible and regulated by TGF-beta regulator. So there are two ways people are looking at. One is a PMP1 as an androgen inducible gene, and PMP1 as regulated by TGF-beta regulated gene. But those are the other cancer contexts like the breast, colon, and lung cancer context. Well, in the prostate, we focused on basically on androgen receptor. And we found this gene is being silenced in prostate cancer, and more specifically, more silenced in Caucasian patients, not in the African American. But the downregulation is similar across the line. So we want to know much more further what is the role of this gene basically and how it's going to control. So we went ahead and start exploring whether it has been the role of TGF-beta or other signaling pathways prostate cancer. So we started investigating into that, and we did the RNA sequencing in the prostate cancer cell line, which is like VCa P, LNCa P cells. And we found there are different isoform presence, which people have already been talking about, but they haven't said very clearly the isoform is specific. So they discussing with Dr. Lee about this, that, hey, this is their very interesting findings that we are looking at different isoforms, but nobody has clearly looked in depth. Is there any isoform specific roles of this gene in prostate context or in other cancer contexts? So we started doing the review of literature, looking for other cancer contexts that people have done. We found that certain people have shown different forms of PMP1, A and B, or 1 and 2 in prostate in other contexts. But literally, when bottom line, they were explaining that only explained the PMP1 or TMP1, transmembrane protein 1. So there's TAG1, solid tumor, there are different names people are calling this gene. So there's whole confusion in the field that somebody called it TMP1, somebody call it TMPAI, somebody will call it as PMP1, somebody call it TAG1. So when we refer to the thing, we don't know which gene we are talking about. So we took that challenge in CPDR that this is our baby, what we call it, because we nurtured this working on almost from 20 years on this gene since we discovered right now. So we that this is our responsibility to put it in a very clear way to the scientific community to understand what PMP1 is exactly. There are so many papers out. So when we are working towards this, we found that there are 5 isoforms. There's a PMP1 and one is that Dr. Lee mentioned that isoform E is a novel which we discovered when we are looking for RNA Seq from TCAJ dataset analysis, even in our in-house data analysis and in VCa P and LNCa P cell lines. We found all 5 isoforms are present in cell lines and prostate tumors. And more especially, there are 2 main isoforms which have been well characterized, which is isoform A and B, which have people already talked about in the prostate and other cancer context. But literally other isoforms, which is C, D, and E, which we have nailed down in this paper, we have been already understudied and not well characterized. So we took this pride to take this study forward. As we know, we are working in the prostate cancer program, so we only focus on in terms of prostate cancer. So we looked into these 3 isoforms, C, D, and E, in the prostate cancer context. And while doing this analysis, when we found out that there is a very great homology between these 3, all the 5 isoforms. All the 5 isoforms bear the same length of the protein, very small amount is different. So we want to know what is the difference between them. So when we did the blast and analyze this protein, we found there's only difference in the N-terminus. So it looks like the function, if any function will be changing here or any regulation is changing, is coming from the N-terminal contact. So when we looked into it and when we identify isoform E, which is a novel isoform which highlighted in this paper, we found that it is the longest N-terminus in the whole isoform. Less C-terminus is highly conserved. And we also went ahead and looked the homology with other species along this. And it looks like it's almost 70 to 80% homology is there with all the species. These are highly conserved genes from the evolution point of view. So which took us more confidence to work into that one. And the biggest question Dr. Lee has always in mind, why there are different role of PMP1 we are seeing in different contexts. So that's key point. He was asking these questions like, hey, we need to sit and look into that, why these same gene is behaving differently in different conditions or in different cancer. So that's challenge we took it. And we started investigating the role of different isoform in at least in two signaling pathways which are already been existing and people are working as a TGF-beta and AR. And we are the first to and able to put it in the way that different PP1 has a different isoforms, and different isoforms has been regulated by different signaling pathways. And it depends on the context dependent and tumor dependent. So this is the very key finding what we were. Now I will give the podium to Dr. Lee to put more in detail, like what his thought process and how we work together towards the consultation of this Oncotarget paper.
I think this manuscript is really a good product for the large discipline team member. So we collaborate with the joint bioinformatics medics team and University of Harvard, and also within CPDR, we also we have a biostatistician really support us. And okay, so I think the biggest challenge is to find a proper serum model to conduct our experiment to validate our hypothesis. But luckily, we start from human samples, so we can get very solid, strong data. We use TCGR dataset. They have almost 500 prostate cancer cases. Yeah. So, and we use their original unprocessed RNA Seq data, so we have very clear idea. Another big challenge is, like as Dr. Sharad mentioned, 5 isoforms, their expression level is different. They have major isoform, minor isoforms, and they have the trivial difference within their gene sequenced and also their amino acid sequenced. So it's very difficult to really tell the difference between these isoforms. And although they have very trivial sequence difference, but their functions different, very different. So, and we must be very careful to process data. So luckily, we get the full access to download the original data, RNA-seq data, so we can really look at that part, and we get very confident data to support their 5 different isoforms, just so we can move ahead. Another thing, we smartly just isolated 2 different contexts, androgen signaling and TGF signaling, and we used SPIC unique siRNA technology to knock down the SPIC gene, and we collaborated with outside company and to get the expression plasmid, so we can express these 2 different isoforms, and we put in different signaling pathway and to look at their functioning. We're lucky to get to that.
And yes, I would also like what he was mentioning, that what is the hardest part when we are designing siRNA, because the sequence difference is so small. But designing a specific primer and probes is very, very challenging when we have to do the validation studies. RNA-seq is good, but to do the validate in the small cohort or validate in the cell lines, the main challenge is to design an isoform-specific primers. And the second, isoform-specific siRNA. I am lucky to have Dr. Lee with me, that he has very great experience in the cancer biology context, and he has the experience from bringing from Danford, which is from breast cancer program. So we work together to streamline how to get it to getting those plasmids and designing the specific siRNAs and overexpression plasmid. I also want to mention here our previous colleagues who is now no more with the CPDR, Dr. Shiv Srivastava and Dr. Alga Swami Srinivasan. They have really the backbone to support this study, and they keep on encouraging us and working toward this goal. And they have very vast experience in the cancer biology and the cell culture models. So we learned a lot from them. And we are taking this gene forward towards a more future as a prognostic and diagnostic market. But that was Dr. Lee's main dream, is that if we can solidly prove that that can be a good prognostic marker, that's what we are looking for the future. But sorry, I interrupted Dr. Lee in between, but he can go ahead with that.
So with this challenge, what we can bring the project to the point, and when you look at the human data, and we really get a good model, can perfectly explain, and how the castration resistance and the metastasis happening. So we know both androgen signaling and beta signaling. The dysfunction of this major signaling is very important to the development and to the instance of the castration resistance and the math. And there are a lot of paper, and our previous research work has shown that. But and how these 5 isoforms come into play as a panel and surrogate to the androgen signaling and T-resin signaling status, and how they navigate the shares and towards the happening of this laser stage. So I just want to, heads up one important thing. So although PNP1E isoform is a very unique isoform, it's the low expression in prostate cancer patient, but they have the 2 very unique attributes. First is responsible to androgen signaling. So brings potential can collaborate with the PNP1B isoform. Second one, PNP1E isoform can function androgen independent prostate cancer series.
That is very key.
This is very, very important. So we know this function of androgen signaling. So it's a very, 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 very essential step when the patient develops resistance to hormone inhibitors. So think about how we can bring back androgen responsiveness to the cells. So make the cells can reverse the cells to the function, inhibited function of the AR inhibitors. This is biggest challenge. So, and think about our collaboration, our interplay between PNP1B isoform and the E isoform. So we found PNP1B isoform, low expression is very common, just mentioned by Dr. Sharad, and lower PNP1 isoform led to activation of androgen signaling because the androgen signaling protein can stabilize. They can also, subsequently they can increase the PNP1E isoform. But the PNP1E isoform, when they start expressing androgen independent cells, prostate cancer cells, they can promote their growth. So we think about this interplay of the 2 different isoforms perfectly can explain why the cells lost expression of androgen receptor, accumulation of androgen receptor, lost response to androgen signaling. They can start growing crazily. So we use these 2 interplay of these isoforms. Potentially we can, if we block this kind of a process they're doing, we potentially can reverse androgen responsiveness of the tumor cells. So we can enhance the sensitivity of the tumor cells, although it's androgen resistance, but it can respond to AR inhibitors. So we think our discovery really can lay a foundation how we can really improve the treatment option, not only the virus marker for the diagnostic marker. We provide another novel insight into the mechanism to how we can increase the sensitivity of tumor cells and reverse responsiveness to AR inhibitors or hormone ablation therapy in cancer genome resistant prostate cancer cells. Here I also want to really thank you for our provost instructor, Dr. Srivastava and Dr. Srinivasan. They really help support us. This project has been last a few years, I think more than 3 years. And eventually, although they have not worked with us anymore, and we really want to thank you their contribution to the project. And also to Center for Prostate Disease co-funding from University of Hair Science, from DHA, and from Provost funding agencies, the NCI. So Dr. Sharad, do you want to talk about what your interest in future?
Yes, so that is very good one, because when you start a study, and as we already mentioned that we are working on this gene and we see the potential, and definitely we want to take it forward. Not only because of androgen receptor, because the interplay of different isoforms that I already mentioned, that how these isoforms interplay each other. Also, for how the interplay of different signaling pathways like AR and TGF-beta working together to make it once become a hormone independent stage, or then TGF-beta signaling pathway can take over, or then another isoform started playing a notorious role in the making a worse disease in the context. So we want to more navigate in that terms. And also, as we mentioned, that PNP1 gene was silenced in prostate cancer. So we want to know whether all the isoforms are really being suppressed in the prostate cancer context, in the cancer and non-cancer context. Also, not only is there any difference with these isoform specific expression in the ethnicity, like we know the Caucasians patients have more aggressive prostate cancer, and we have seen that different isoforms have been navigating to a more aggressive context of the prostate cancer with early stages and later stages. That's what our plan. We want to look whether within what stage of the prostate cancer, which isoforms are playing a key role. And that's what Dr. Lee's main review is to look how do we can monitor the progression with these isoforms. And based on these isoforms, specific how we can treat the patient depending on the role of these isoforms in which stage they are. We want to say that there is no one therapy will be there. There are different kind of stages, different kind of genetic makeup, different kind of regulations are playing a role, because as we know that prostate cancer is very heterogeneous and it's not easy to treat that patient. So we need to monitor the patient, and where I feel or we feel together that these isoform specific roles in the context or in the cellular context, in a disease-specific context, will play a very great role, not only the monitoring, but also in the therapeutic options. So our future is to look how to develop inhibitors for TGF-beta or AR inhibitors to stop the progression of the prostate cancer, and also to monitor the progression of the patient and treat in the timely manner. So that's the main focus we want to look into that future.
Yeah, so I just give a little bit more. So for our group, we currently really want to dissect underlying biology of the castration resistance prostate cancer and the MEDS prostate cancer. There's a major challenge. We have repeated so many times that one. And so we really want to explore the two possibilities of the PNP1 forms. One with the PNP1 isoforms as a panel biomarker for indicating the disease progression and treatment options. So think about if the patients have large expression of PNP, the potential to develop resistance to hormone in ablation surrogate.
That's true.
Or they have higher expression of PNP1E, maybe they have higher chance to develop MEDS or hormone resistance.
Yes.
So although we have some preliminary data from TCGA, but Center for Prostate Disease Research, they have the more comprehensive cohort and to cover MEDS, to cover castration resistance and very indolent prostate disease. So we proposed to further use the more accurate technology, including digital PCR or nanostring, can really look at this 5 isoform expression, transcript level in these castration-resistant and MEDS prostate cancer patients, and also really look at their correlation with disease progression, include Gleason score, nuclear grading, and pathology stages, BCR and MEDS, and also overall survival rate. So this is the thing we really want to look at.
Right.
And also we want to explore how these isoforms can inhibit both androgen receptor signaling and TGF-beta signaling through the proteasome-dependent pathway. It's different from direct block binding between the receptors and ligands. More deeply, we found actually they caused the degradation, ubiquination, and degradation of androgen receptor or TGF-beta, or TGF-beta mediated includes that family. So this is another major challenge. Based on that, we want to use more look at their crystal protein structure and the binding sites. We already have some very beautiful preliminary data. Hopefully, we also can publish in soon. Oncotarget, and we found actually their binding to this the binding between these isoforms and the androgen receptor, TGF-beta receptors, they're not universal, they're very unique. So for the indicators, they're just navigate the cells through different signal pathway. So based on that, we want to design more specific AR or TGF-beta inhibitors, signal inhibitors, and they can either bind with other classical AR inhibitors, work in more efficient way, or they can function through different signal pathway. So we want to explore two major angles, biomarker for progression monitoring or therapeutic angle. to see whether they can inhibit, block AR signal and TWL, especially in later stage prostate cancer.
Lethal prostate cancer, like NCRPC, castration prostate cancer, or bone metastasis, or the patient what we are looking for, who are recurred biochemical recurred patient. That is also the field is now when we clarified that there are isoform specific roles in the prostate cancer. It will be a good time for other investigators to look into in other cancer contexts. What is these isoforms are really playing a similar role or different role in those cancers, or their aggressiveness types and everything, which is very important to look into that one. And also not only the androgen, because it is still certain isoforms, a role is not so clear about AR and TGF-beta signaling. Maybe there are involvement of other signaling pathways need to be investigated. That is our also plan to look if any novel signaling pathways is also navigating in different, or maybe in the latest stage where AR signaling is gone or where TGF-beta signaling is not working. Maybe some other signaling pathways is navigating to making the worst disease or recur the patient. So that is...
Yeah, we have some candidates. Like
yes, right. I'm glad you mentioned this one. Yes.
So we were planning to... we think the PMP1 is really multifunction protein and is really playing important role in breast cancer, but also in our other hormone related like breast cancer and also some lung cancer. So there's so many. Think about the androgen signaling and TGF-beta signaling. It's very universal to a lot of different tissue and organs. And think about our strong regulation and mechanism behind this. So we're really confident. We're happy to collaborate with the other researchers from other solid tumors, especially for lung, breast, and colon cancer. We can first explore their origin and to see whether we really can contribute to the cure of the lethal breast cancer field. And we try to continue to work and to NCI with NCI and DoD funding. And hopefully we have some paper can publish soon.
Yes, and I cannot believe that he is really expert in the laser capture microdissection. That is the key of all of our research, why we are able to produce such a clean data, because he very specifically isolated the clean cells from only tumor-specific cells, and we also can distribute the cells in normal and tumor-adjacent tumors. So instead of we are mixing all the tumor, like just cutting, maybe he can explain more in that, we are specifically isolating a single cells from the cancer and the normal, close benign and normal cells. And then we are comparing, that's how we got a very clear expression of these isoforms when we are validating in our studies. That's how beautifully he has done that.
Yeah, these are data set, but we use technology.
Right.
Luckily, we have laser capture technology on board, so we can utilize to really dissect the cleaning. Our purity, sample purity is over 90%. Right. So, and the tumor heterogeneity is another big challenge.
Very big challenge.
So we can use either you can use the in situ technology, include IPC, or in situ hybridization technology to look at the individual cells. Right. Or either you can laser capture, so we can really dissect the specific tumor and separate from non-tumor. So we get a clean data. You get a cleaner data, you have more confidence and you have stronger statement.
Yes. That's how the plan is how to move forward with this technology, that how we can validate the silencing and epigenetic mechanism of our RNA methylation with this to move forward with this and with the different ethnicity and different backgrounds. So yes, thank you. And
thank you, Ryan. We think we are good. So